For most of human history, we assumed dinosaurs looked like lizards. Gray-green, brown dulls. Because that's what made sense. They were reptiles. Reptiles have dull colors, case closed. For over a century, every museum reconstruction, every textbook illustration, every movie monster followed that assumption. And the strangest part, the evidence that would prove all of it wrong had been sitting in museums for decades. In drawers, on shelves, already collected, already preserved. Nobody had thought to look at it the right way. Then in 2008, a scientist looked at a fossil feather under an electron microscope and found something that changed everything. Tiny capsules preserved inside the rock, identical in shape and arrangement to the structures that produce color in modern birds. And when he cross-referenced those capsule shapes against living species, he could read the color directly from the fossil. Black feathers, red feathers, white feathers. Within 2 years, scientists had reconstructed the full-body color of a dinosaur that died 160 million years ago. Not guesswork, not imagination. Direct evidence written in stone. And what those fossils revealed wasn't gray, wasn't green, wasn't dull. One dinosaur had a brilliant red crest. Another had glossy iridescent black feathers that shimmered like a raven's wing. A third wore a dark mask across its face and a striped tail designed to disappear into open terrain. And a long-necked giant we had been drawing as plain gray for 100 years may have had color patterns we are only now beginning to understand. The story of how scientists cracked the color code of dinosaurs is one of the most remarkable detective stories in the history of science. And it starts not in a dig site, but in a laboratory with a question nobody had thought to ask. For decades, paleontologists studied fossil feathers for one thing: structure, shape, the physical architecture that helped them understand evolution and the origins of flight. Color never entered the conversation. Because everyone assumed color couldn't survive fossilization. Organic pigments decay, they break down, they vanish. So the assumption was clean and simple: whatever colors these animals had in life, the fossil record would never tell us. That assumption was wrong. In 2008, a researcher named Jakob Winther was examining fossil feathers under an electron microscope at Yale University. Not looking for color, looking at structure. And he noticed something unusual. Tiny elongated capsules embedded in the feather, hundreds of them arranged in patterns. He recognized them immediately. Melanosomes, microscopic organelles found inside the cells of skin, hair, and feathers. They contain melanin, the pigment responsible for black, brown, gray, and reddish tones across the animal kingdom. Every feather you have ever seen on a bird outside your window carries these same structures. And in modern birds, the shape of melanosomes directly predicts color. Spherical melanosomes produce reddish-brown tones. Elongated rod-shaped melanosomes produce black. Flat platelet-shaped melanosomes create iridescent shimmer. Winther realized something that stopped him mid-analysis. If melanosomes preserve their shape in fossils, and if shape predicts color, then you could read the color of an extinct animal directly from its fossil. Not infer it, not guess it, read it. For paleontology, that idea was almost dangerous. And then the consequence of that realization landed. If he was right, every dinosaur reconstruction ever made could be wrong. Every museum mural, every textbook illustration, every gray-green monster that had defined our image of the prehistoric world for over a century. All of it built on an assumption that science had never actually tested. His paper, published in Biology Letters in 2008, was 5 pages long, but its implications stretched back 165 million years. Fossil feathers are preserved as melanosomes. Their distribution and shape can preserve the original color patterns of the animal. The field took notice. And within 2 years, a team in China would use that discovery to do something nobody had ever done before. What does a 160-million-year-old dinosaur actually look like in color? The fossil had been in a Chinese museum collection for years. A small feathered dinosaur from the late Jurassic, about the size of a pigeon, 4 wings, covered in well-preserved feathers studied extensively for their structure. In 2010, a team led by Guo Li decided to do something no one had attempted before. They mapped every single feather on the body under an electron microscope. Not a sample, not a region, every feather across the entire animal. They extracted tiny samples from dozens of locations, mapped the melanosome shapes at each point, cross-referenced every result against a database of modern bird feather colors built from hundreds of living species. And then they assembled the picture. The body was predominantly black and white, dark wing feathers, gray tones across the back and tail, the kind of pattern you might see on a magpie today. But the head told a completely different story. A reddish-brown speckled crown, dense rufous coloring concentrated at the top of the skull, an actual crest of feathers flushed with warm red pigmentation. Think of a woodpecker, that bold red cap that makes it instantly recognizable from 30 meters away. This dinosaur had something functionally similar, a visual signal, a display structure, a marker of identity or sexual fitness visible to every other member of its species. This wasn't a gray lizard. This was a visually sophisticated animal using color exactly the way modern birds use color. For the first time in history, humanity wasn't imagining a dinosaur. It was seeing one. The paper published in Science in 2010 established something permanent. Melanosome analysis worked. Color reconstruction from fossils was real, not theoretical, not speculative, demonstrated. And it immediately raised a much bigger question. If one feathered dinosaur had complex color patterns, how many others did? How sophisticated could those colors get? The next discovery had an answer nobody anticipated. Because the next dinosaur didn't just have color, it had something far stranger. 2 years after the first full-color reconstruction, the same research team published another paper. Same journal, same method, different dinosaur, and a completely different result. The subject was a four-winged predator from early Cretaceous China, slightly larger than the previous specimen. A fast, agile hunter that likely used elevated terrain and dense vegetation to launch attacks from above. When they mapped the melanosomes across its preserved feathers, they didn't find the simple rod shapes that indicate flat black. They didn't find spherical forms indicating reddish tones. They found platelets, thin, flattened melanosomes arranged in stacked layers. In modern birds, that specific arrangement produces one result: iridescence. The structural shimmer you see in a raven catching direct sunlight, colors that shift depending on viewing angle, black that becomes blue-green as the light changes, darkness that contains brilliance. In sunlight, the feathers of this dinosaur wouldn't have looked black at all. They would have flashed blue and green as it moved. A predator that shimmered. The team's analysis, published in Science in 2012, concluded that this animal had glossy iridescent plumage across its entire body. The same optical architecture that makes starlings and hummingbirds among the most visually striking animals alive today. And that tells us something critical. Iridescent plumage in modern birds almost universally serves one purpose: display. It signals genetic quality, health, the ability to grow and maintain expensive optical structures that only a genuinely healthy animal can produce. Which means this wasn't just a predator. It was an animal engaged in the same kind of mate competition that drives peacocks to grow 2-meter tails. Color wasn't decoration, it decided who reproduced. And who disappeared. The evolutionary thread connecting these feathered hunters to modern birds wasn't just about feathers and flight. It was about the social and sexual logic of color itself. But color doesn't only signal, sometimes it hides. And the next discovery revealed something even more sophisticated than display. Because some dinosaurs weren't trying to be seen, they were trying to vanish. 2017. A team led by Fian Smithwick at the University of Bristol published a paper in Current Biology that took the science of dinosaur color in a completely different direction. Not display, not iridescence. Camouflage. The subject was a small feathered predator from early Cretaceous China, turkey-sized, fast, one of the first feathered dinosaurs ever discovered when its fossils emerged in the 1990s. Smithwick's team applied melanosome analysis across multiple well-preserved specimens, and what they reconstructed was a color pattern of extraordinary biological precision. A banded tail, alternating light and dark rings running down its length, like a ringtail lemur or a raccoon today. A pale underbelly with progressively darker coloring toward the back. The classic countershading pattern that makes an animal harder to detect against both sky and ground simultaneously. And across the face, a dark band running through the eyes, the pattern we call a bandit mask, seen today in raccoons, badgers, and certain hawk species. You see the same strategy everywhere in the modern world. Foxes, deer, even military camouflage borrows from the same principles. Different time period, same survival logic. But here is where the science becomes genuinely remarkable. The team didn't just reconstruct the colors. They used the color pattern itself as ecological evidence. Because countershading isn't random. The precise position of the boundary between dark and light on the body encodes information about the lighting environment the animal evolved in. In dense forest where sunlight is diffuse and scattered, countershading boundaries sit lower on the body. In open terrain where sunlight is direct and overhead, those boundaries shift higher. When Smithwick's team mapped the countershading boundary on this dinosaur, it matched the signature of an animal living in open, well-lit habitat, not deep forest. They had used preserved color patterns in a 130-million-year-old fossil to reconstruct the environment the animal hunted in. Not from geology, not from plant fossils. From the animal's own camouflage. The dark eye mask added another layer. In modern animals living in exposed environments, masks reduce glare from bright overhead sunlight and improve visual precision in high-contrast conditions. This small predator, hunting in open terrain, camouflaged against a bright sky, with a striped tail that broke up its outline against dry vegetation and rock. Not a monster, a precision-engineered hunting machine built by evolution to disappear in plain sight. And the fact that we know this, not from imagination but from preserved pigment structures in a 130-million-year-old fossil, is one of the most extraordinary things science has ever achieved. But the achievement almost fell apart entirely. Because scientists then realized something deeply unsettling. What if they had been wrong all along? The melanosome method was elegant, powerful, and it rested on one central assumption. Melanosome shape predicts color, that relationship established from studying hundreds of modern bird species. Thousands of feather samples, peer-reviewed, replicated, published. And then, in 2013, a team at the University of Texas raised a challenge that sent a tremor through the entire field. Their concern was contamination. Fossilization doesn't happen in a sterile environment. When an animal dies, bacteria colonize the decaying tissue within hours. And some bacteria, they argued, form microscopic rod-shaped structures that look remarkably similar to melanosomes under an electron microscope. If you couldn't reliably distinguish fossilized melanosomes from bacterial remnants, how confident could you be in any color reconstruction? If the critics were right, the entire field could collapse overnight. Every color reconstruction, every red crest, every iridescent shimmer, every camouflage pattern. All of it potentially built on misidentified bacteria. The response from the field was not dismissal, it was science. Researchers developed new methods to chemically test fossil samples directly for melanin chemistry rather than relying on shape alone. Mass spectrometry, secondary ion mass spectrometry, techniques that can detect the actual organic chemistry of melanin even in fossils hundreds of millions of years old. The results came back consistent. Melanin chemistry is detectable in fossil specimens. The structures identified as melanosomes do contain melanin. The bacterial contamination concern, while scientifically legitimate and worth testing rigorously, does not invalidate well-controlled reconstructions. The fossils were speaking, and the signal was real. But the debate did something valuable that pure discovery never could. It forced the field to be honest. Jakob Vinther himself, reviewing the paleocolor field in 2015, was explicit about the limits. Melanin survives, its chemistry is detectable, its structure is readable, but every reconstruction carries uncertainty. The good scientists say so clearly. The color patterns revealed by these fossils are not definitive portraits. They are the most rigorous scientific estimates ever made about what extinct animals actually looked like. That distinction matters, because the moment science stops acknowledging uncertainty, it stops being science. And this field, tested by genuine challenge, emerged stronger. Which gave researchers the confidence to ask a question that seemed almost impossible. What about the giants, the animals so large that color seems almost irrelevant? December 2025, a paper in Royal Society Open Science changed the conversation again. Not about feathered hunters this time, not about bird-like predators with display crests and iridescent wings. About sauropods, the long-necked giants, animals that stretch 25 meters from nose to tail, creatures so large that a human standing beneath one would have felt the ground shake with each footstep. Not a small feathered hunter this time, one of the largest animals that ever walked on land. For a century, we had been drawing these animals as plain gray, not because of evidence, because of assumption, Their skin seemed too thick, too simple, too basic for complex pigmentation. A team of researchers, including Glenn Storrs from the Cincinnati Museum Center, analyzed fossil skin samples from juvenile specimens of one of these giants. Not feathers, scales, tiny epidermal scales from the skin surface. And they found melanosomes, in the last place anyone expected. Not one shape, multiple shapes. Spherical, elongated, and intermediate forms distributed differently across separate body regions. Which means this animal didn't just have one color. It had the biological architecture for a range of colors across different parts of its body. The gray giants that had filled museum halls for over 100 years suddenly felt unfinished. The researchers were appropriately careful. The paper is explicit that this is the first evidence, not a complete reconstruction. Sample size is limited, further study is needed. The science demands that caution. But the implication cannot be avoided. An animal with diverse melanosome shapes across different body regions was producing different pigments in different areas. That means patterning. That means variation. That means the possibility of markings, countershading, color gradients, or patterns we have not yet decoded. Think about every children's book you have ever seen with a plain gray long-necked dinosaur standing in a swamp. Think about every museum illustration, every documentary reconstruction. The science is now suggesting those images are probably wrong, not slightly wrong. Fundamentally wrong. Because color is not decoration. It is biology. It encodes how animals survive, how they attract mates, how they warn predators, how they recognize their own species. If these giants had the melanosome architecture for complex color, they were using it for something. We just don't yet know what. And that question, hanging over one of the most familiar images in all of paleontology, is exactly where this field stands today. So what does all of this actually mean for how we understand these animals? Let's go back to that first assumption. Gray-green-brown dulls. That assumption wasn't based on evidence. It was based on the absence of evidence. Nobody could read color from fossils, so they filled the gap with the nearest living analog they knew, reptiles, and many reptiles are dull. But dinosaurs weren't simply reptiles. They were a separate lineage with a separate evolutionary history spanning 165 million years. And for a significant part of that history, they were covered in feathers, scales, and skin structures sophisticated enough to produce some of the most complex visual signals in the animal kingdom. We know this now not from imagination, from melanosomes. From a structure smaller than a human red blood cell, preserved in ancient rock, whose shape still carries the chemical signature of pigment that pumped color through living tissue 160 million years ago. One dinosaur had a red crest and blackened white patterned wings. Another shimmered with iridescent blue-black plumage in direct sunlight. A third wore a bandit mask and a striped tail, precisely camouflaged for hunting in open terrain. And a giant we thought we understood may have carried color patterns across its skin that we haven't yet learned to read. The world of the dinosaurs wasn't a world of stone. It was a world of color. Each of these discoveries didn't just change what those animals looked like, it changed what they were. An animal with a display crest is competing for mates. An animal with iridescent plumage is advertising genetic quality. An animal with countershading camouflage is navigating predator pressure from multiple directions. An animal with diverse melanosome architecture is communicating something to its world. These are not monsters. They are not dim-witted giants frozen in gray museum dioramas. They are sophisticated biological entities doing exactly what every successful species does. Surviving, competing, reproducing, communicating. In color. And we almost missed all of it. For 100 years we walked past these fossils assuming color was gone, that the stone held shape but not pigment, structure but not signal. It took one scientist looking at something everyone else had already studied, and asking a question so simple it seems almost embarrassing in retrospect. What are those tiny capsules and what do they mean? That question launched a field, gave us red crests and iridescent wings and striped tails, and now the first evidence of color in the giants themselves. And it leaves one final thought worth sitting with. If we were wrong about color for 100 years, what else are we wrong about? What other signals are written into these stones, waiting for someone to ask the right question? The fossils haven't changed. We just keep getting better at listening to them.